Hello guys. Today we are going to discuss about a blood test. It is called BCR ABL Fusion. Use of the test to help diagnose and monitor the treatment of chronic myeloid leukemia. It's a cancer and a type of acute lymphoblastic leukemia. Two types of cancers, CML, ALL. BCR-ABL refer to a gene sequence found in an abnormal chromosome 22 of some people with certain forms of leukemia. Humans normally have 23 pairs of chromosomes, including 22 pairs of non-sex determining chromosomes and one pair of sex chromosomes. Chromosomes contain a person's inherited genetic information. The genes that resides there form the blueprint for the production of countless proteins. Sometimes changes can occur to a person's chromosomes and genes during their lifetime because of exposure to radiation, toxins, and unknown regions. The BCR-ABL gene sequence is one such acquired change that is formed when pieces of chromosome 9 and chromosome 22 break off and switch places. When this occurs, the ABL region in chromosome 9 fuses with BCR gene region in chromosome 22. This is referred to as reciprocal translocation, and this particular one is commonly expressed as 9 is to 22. A derivative chromosome 22 that has the BCR-ABL gene sequence is known as Philadelphia (PH) chromosome. It is strongly associated with the chronic myeloid leukemia (CML), less commonly with one type of acute lymphoblastic leukemia, and rarely with acute lymphoid leukemia. At present, the diagnosis around 90 to 95% of cases of CML are found to have Philadelphia chromosome. All cases have BCR-ABL fusion gene, including the Philadelphia chromosome negative case. In this small fraction of a cases, the gene have fused one on a more macroscopic scale, not involving large chromosomal segment. The BCR-ABL gene sequence at the fusion site encodes an abnormal protein. The abnormal BCR-ABL protein is a tyrosine kinase enzyme, a normal signaling protein which has become permanently switched on and is responsible for the development of CML and a type of ALL. When large number of abnormal leukemic cells started to crowd out the normal blood cell precursor in the bone marrow, signs and symptoms of leukemia started to emerge. Treatment of these leukemias typically involve a tyrosine kinase inhibitor given as daily tablet. Testing for BCL-ABL detect the PH chromosome and BCR-ABL fusion gene or its transcript. The RNA copies made by the cell from the abnormal stretches of DNA. The presence of BCL-ABL abnormality confirm the clinical diagnosis in CML or a type of ALL. These are distinguished by the typical appearance of the cells through microscopic and in case of ALL by flow cytometry, which looks at a molecules expressed inside the cell or more commonly expressed as the leukemic cell surfaces. There are several different types of BCR-ABL tests available, including cytogenetics, chromosome analysis, or simply we can call karyotyping. This test looks at a chromosome under a microscope to detect structural or numerical abnormalities. Cells in a shape of a blood or a bone marrow are grown in a laboratory and then examined to determine if the Philadelphia chromosome is present. Other chromosomal abnormalities can also be detected. The second technique is FISH, fluorescence in situ hybridization. This test method uses differently colored fluorescent dye labeled probes to light up the BCR and ABL gene sequences. Cells are examined under a microscope to determine the proportion, if any, where the colored dots are fused together, indicating BCR ABL translocation. The third one is genetic molecular testing, qualitative or quantitative. The polymerase chain reaction based qualitative and quantitative tests detect and measure BCR-ABL gene transcripts or gene product unit in a patient's blood, bone marrow samples. This is the most sensitive test for BCR-ABL and is used to diagnose the minority of cases, negative for Philadelphia chromosome. It is also used to check the presence to tyrosine kinase inhibitor. Variant mutations of BCR-ABL can also be analyzed if indicated. These mutations are responsible for treatment resistance to TKI. If the response to treatment is suboptimal, mutational analysis may tell your doctor which TKI will be more effective. The sample required. A blood sample is obtained by inserting a needle into a vein in the arm, or a bone marrow sample is collected using a bone marrow aspiration or sometimes biopsy procedure. Results: if a person has abnormal white blood cells in a bone marrow and has the Philadelphia chromosome and BCR-ABL gene sequence, then that person has CML or Ph positive all. Of these who have CML, 90 to 95% have the Ph chromosome by cytogenetics and 100% have BCR-ABL gene by FISH or qualitative BCR-ABL molecular testing. About 25% of the adult all and 3% of the childhood all are positive for Ph chromosome and BCR-ABL gene sequence. That is about today's testing.